Carcinogens. Carcinogenesis, also called oncogenesis or tumorigenesis, is the formation of cancer whereby normal cells are transformed into cancer cells. The process is characterized by changes at the cellular, genetic, and epigenetic levels and abnormal cell division. Cell division is a physiological process that occurs in almost all tissues and under a variety of circumstances. Normally, the balance between proliferation and programmed cell death in the form of apoptosis is maintained to ensure the integrity of tissues and organs. According to the prevailing accepted theory of carcinogenesis, the somatic mutation theory, mutation in DNA and epimutations that led to cancer disrupt these orderly processes by disrupting the programming, regulating the processes, upsetting the normal balance between proliferation and cell death. This results in uncontrolled cell division and the evolution of those cells by natural selection in the body. Only certain mutations lead to cancer, whereas the majority of mutations do not. Cancers and tumors are caused by a series of mutations. Each mutation alters the behavior of the cell somewhat. Cancer is fundamentally a disease of regulation of tissue growth in order for a normal cell to transform into cancer cells. Genes that regulate cell growth and differentiation must be altered. Genetic and epigenetic changes can occur at many levels, from gain or loss of entire chromosomes to a mutation affecting a single DNA nucleotide, or to silencing or activating a microRNA that controls expression of 100 to 500 genes. There are two broad categories of genes that are affected by these changes. Oncogenesis may be normal genes that are expressed at inappropriately high levels or altered genes that have novel properties. In either case, expression of these genes promotes the malignant phenotype of cancer cells. Tumor suppressor genes are genes that inhibit cell division, survival, or other properties of cancer cells. Tumor suppressor genes are often disabled or by cancer promoting genetic changes. Finally, oncovirinae viruses that contain an oncogene are categorized as oncogenic because they trigger the growth of tumorous tissue in the host. This process is also referred to as viral transformation. The causes: genetic and epigenetic causes. There is a diverse classification scheme for various genomic changes that may contribute to generation of cancer cells. Many of these changes are mutations or changes in the nucleotide sequences of genomic DNA. There are also many epigenetic changes that alter whether genes are expressed or not expressed. Aneuploidy, the presence of an abnormal number of chromosomes, is one genomic change that is not a mutation and may involve either gain or loss of one or more chromosomes through errors in mitosis. Large-scale mutations involve either the deletion or duplication of a portion of a chromosome. Genomic amplification occurs when a cell gains many copies, often 20 or more copies, of a small chromosomal region, usually containing one or more oncogenes and adjacent genetic material. Translocation occurs when two separate chromosomal regions become abnormally fused, often at a characteristic location. A well-known example of this is Philadelphia chromosome or translocation of chromosomes of 9 and 22, which occurs in chronic myelogenous leukemia and results in production of BCR absorption protein and oncogenic tyrosine kinase. Small-scale mutations include point mutation, deletion, and insertion, which may occur in the promoter of gene and affects its expression, or maybe occur in the gene's coding sequence and alter the function or stability of its protein product. Disruption of a single gene may also result from integration of genomic material from DNA virus or retrovirus, and such an event may also result in the expression of viral oncogenes in the affected cells and its descendants.